waters that have been out of contact with the surface for the longest. The deeper waters have had more recent surface contact.
Yeah, kind of those oxygen differences are kind of a combination of oxygenated waters coming up and then older waters having had a lot of respiration from all these.
And surface productivity has a big effect on that. The sinking and decay of phytoplankton.
Oh, there's a squid.
Cool.
Hi.
Hey, squid.
Squid.
I feel like they usually swim by so much faster.
Squid.
That was nice.
I'm meeting you at 090.
Yep, that's correct.
Almost at 045.
I feel like my day can never really start until we see that seafloor. You can have your breakfast and your coffee, but just until you see the seafloor, it's a lot of anticipation.
There we are. Thanks, Kylie.
Getting there slowly.
All right. Shall we mosey on down?
Yeah, let's go.
Can you see the seafloor?
We're coming down together to see where
We're about 32 meters off bottom.
So
just getting themselves aligned.
Usually it's easier to do without any landmark features in the way, even though we'll be on flat and bottom. Lisa, how high up would you like the Niskin to be pulled? Around 2 meters off or something?
yeah, that's
higher?
fine. That's great.
Roger that.
Let's just confirm we're over horrible sediments. Although we have enough Niskins on board that it isn't, we can take another.
Raj. All right, we're starting to see.
Got a visual.
7 meters off now.
Sediment.
That looks like sediment.
We got sediment.
And a fish.
All right. Is this an appropriate altitude for you? It's about 3. Is that all right?
I think that works.
Raj.
Yep.
3 meters off bottom.
Kylie, want to turn on the porch light, please?
Yes, please.
Thank you.
No No problem.
Anyone follow me in on the bubble there, please?
Yeah.
Aim in for number 1.
Yes, please.
Roger that. Swing and a miss. Oh,
Can you remind me how far we are off bottom, Aaron?
4 meters.
4 meters.
All right, thank you.
My auto XY went all weird.
That did go a bit weird, did it?
Yeah.
I did not press the button, I promise.
Roger that. Popped. All right.
All right, that was NA124
Thank you.
Roger that. All right. Kylie, I'm going to come back a little towards you. I don't know why, but I think it went a little bit on a runaway there.
Yeah, we're pretty stretched out. I got tugged.
Raj.
We're low on delta, but because you're so far out in front,
Yeah.
I come up whenever. That
Right.
makes sense too.
Roger that.
Kylie, are we okay for now? I can back up when we're settled if need be. Yeah. Yeah. Okay. I found you. Found him? Yeah.
All right. And anywhere around here, we'll land, set down, and take some cores, and we can do our light white, black and white balance there. That sound good with you, Lisa?
Yep. Thank you.
Raj.
I don't know if you need to do a white balance or.
Oh, stand by.
Oh, what is that underneath?
Fishing gear underneath us. Nice.
Oh, hooray.
I don't see anything in the camera, though.
Maybe kelp?
What's that?
Or you think it's gear?
I think it's
Some
gear.
kelp?
Go ahead and push on in there, Al.
Is that an enemy on it?
Yeah.
Okay. Well, don't sit down on that. It's
Rubber. What is that? Some tubing.
a connector.
Yeah, it looks like some kind of cable connector.
Okay.
Interesting.
Full wide. Well, let's not sit down there. Let's make sure we're clear.
At that table, like rock.
A rock.
Kind of one random rock in the middle of nowhere.
Yeah, there's some other rocks possibly.
I guess I was looking at Herc and it looked practically like the moon.
Oh, that's cool.
There's a tulip sponge. Hylanema. A nice view. It's anemones. So we're getting ready to take a series of push cores. Gabe, you want to talk about what they'll be used for?
Yeah, so from the sediment, we'll be looking at the bacteria that live in these sediments. And we've been collecting sediments from all dive sites, almost all except one that we did not find sediment. but we'll look into comparing the different communities of bacteria. We'll use a combination of techniques to extract DNA. From these sediments, we could extract also RNA, and we could directly use this sediment to plate on the lab and grow the different microbes found from these sediments, and kind of like get a sense of what is the community and how different are these communities from what has been already discovered, and from the ones that we do end up studying, we could then try to figure out what is their potential to produce medicinally important
everything good in
compounds.
your starboard side? I mean, yeah, yeah.
Yeah.
Just duster stuff.
Yeah, it's just the verts right now.
Okay.
Very cool.
Oh, because you're floaty and you're parked.
Yeah.
So it's working hard.
Working something. All right.
Am I okay to reset?
Yeah, go ahead.
Kylie, I'm going to block your camera now on Zeus.
Okay.
So we're looking at a Panicia sea cucumber. They've been around for quite a few of the dives.
Yeah.
All right, Kylie, go ahead and switch over camera. Oh, switch over salvos.
To sample?
I feel like we've seen dark purple, light purple, and clear
All right.
sea cucumbers since we've been diving in this other California borderland.
Go ahead and pop out the starboard, please.
You got it.
And Lisa, are we going to take all 5 here?
Sample tray out.
Yes,
Gotcha.
all right next to each other. But are we doing only 5
Yes,
today?
5. Yeah, all 5.
Ooh, oh, scared me. Gosh.
Oh, from the marker?
Yeah.
I wanted that to happen on Halloween.
I had a repeat of the sea cucumber flying out, so I was like, no.
Oh, we should totally draw a face on it. Pop up.
I thought you guys were going to put one of your pumpkins on board.
Oh, we still need to carve those, actually. That's a
Soon.
But if you put a pumpkin on in there, would it like split under pressure?
It would probably float away, from what I've heard at least.
We've done a vegetable test. We've taken down all kinds of vegetables to see how they function under pressure.
And would they?
And only the green pepper squished a little, but most of them don't.
Interesting.
We did a pumpkin 2018 Halloween, a mini one, like a little gourd.
Oh, that's right. Yeah, we strapped that to the porch, yeah.
He just got soft.
I guess they do, fruits and vegetables do have a pretty high water content, so it's probably hard to compress.
I remember when I
water.
was diving, when I was getting certified, our instructor had an orange and he showed it to us at the surface. Then once we reached the bottom, he pulled it out of his pocket and it was like just an inch big.
Nice.
So we're like, oh wow, it compressed and whatnot.
That he had taken a tangerine.
Yeah,
down.
so he had a little cutie in his other pocket. So then he showed that to us and we didn't even think of it at first.
All right, Kylie, go ahead.
What a
Switch
trickster.
over to dive mode.
Well, I do have some styrofoam cups on board. We haven't.
I was
put
just
any on
gonna
her.
say that.
I don't know. Do you guys do that with
Yeah, we still do the
Argus,
cups.
maybe?
Indeed. Guess we haven't really put them out this year because we haven't had too many science communication fellows out or people that have requested them.
Maybe our first time cruisers.
Yeah, I like
should
doing
do it.
that. Usually we put it on the CTV.
All right, is around here good for you guys?
Yes.
Only if it's deep.
Only if it's deep, Raj.
Just
I think it will be.
it will be.
Good thoughts.
Yeah, it was interesting in some of the sandy cores from the second site yesterday. The Jensen group found some small fragments of phosphorite in their cores. So either an interesting thought.
Was it
Yesterday, yeah. The second site, which was a little bit shallower.
Oh, okay. That's what you guys were hoping for, Paul was saying. He thought there might be some
Yeah.
fragments.
I'll definitely have to investigate. Could just be broken up pieces like me.
That looks like a pretty good core.
Yep.
All right, go ahead, Kylie. Ooh. Getting a thorough dusting back here, aren't we? All right, Kylie, go ahead. Oh, I already moved myself. All right.
We're always so quiet during these corings.
I know. Well, I figure that
Too
takes a
focused.
lot of
We
concentration.
don't want to disturb.
I don't know if anybody home can see the sea state in any of the views, but it is just perfect weather out here these past couple days. So calm.
I don't know if I've ever seen such a stretch of like perfect
It's so
weather.
amazing. Last night I went up on the observation deck at about 1 a.m. after finishing in the lab, and it was just moonlight and super calm.
Okay.
I felt like I was in an old-timey painting where they just kind of paint the water like grayish, smooth. It was,
I've seen it quite a bit, but often further south.
yeah, for these calm waters. Lisa's good luck.
Let's do that, actually.
Yeah, I think most people think, oh, you guys are out on a boat. You must get to swim and do some sunbathing. But turns out a lot of time in the dark ROV room or working in the labs.
Yeah, sometimes you have to remind yourself to go outside.
Yeah. Get a little sunlight.
Yeah, it's not all mimosas and jacuzzis.
It's super fun, but in a different way.
My brother thought that's what I would be doing.
Oh, the high
When I
voltage
say, like,
cabinet went down
research
again.
cruise, he's like, oh, like, what, you're going to be gambling,
Go ahead, Kylie.
drinking? Not quite.
When my kids were little and I was out on a cruise, their class was allowed to email us questions. And we got questions about, do we have an orchestra on board? Pay for us.
Just a bunch of nerds.
We have an invertebrate orchestra.
Yeah, that's true.
We lost water cam, I see.
Oh, sorry, I wire cam. I guess it's on the sample salvo, so. Yeah, get in there.
So I noticed that for both groups handling the cores after we get them on deck, we've been subsectioning based on depth within the core. Is that just for ease of sampling, or do you expect to find different communities at different depths, and what are the reasons for those depth distributions of different communities?
Go ahead, Kylie.
Anybody who handles cores?
Well, for the bigger animals that we study, they are zoned by depth, but it's also easier to sort them under a microscope when the samples are smaller, rather than one big hunk of mud. But most of the animals are concentrated near the surface, and as you go deeper in the sediment, you get different lifestyles, and also the bigger animals live deeper because they often need to make a connection to the surface sediment water interface, and so the bigger ones can go up and down a tube or a burrow to do that. So usually below 5 centimeters we tend to find the biggest animals, but very few of them.
Very cool. And so those changes with depth are due to availability of organic carbon to eat, or is it due to oxygen and different metals redox?
All of that changes down core. But I want to talk about the bacteria. Okay.
And is that the same for the microbes?
Can't see.
I think I'll recruit Paul or Caitlin to answer that. I'm not sure about the microbes. Yeah,
the microbes change down core because the geochemistry changes. So they're different.
then I wouldn't be able to sit down that much.
All those factors.
I'm gonna turn down my jogging now a little bit.
Will influence the bacterial.
I think it's partially that floaty thing there. It's changing up the auto exposure on the camera.
Yeah.
I had a question
reflective
on my chat from the side who wants to know why the cores have to stay in so long. For those people watching, it's simply a way to keep moving the sediment, because pulling them out creates a disturbance locally. By putting them all in first, we know that each one's intact.
Yeah.
They're not incubating or anything like that out there.
Yeah. No extra process is going on. I think it's also efficient for the pilot, ROV pilot, who's working on the arm, to put them all out in a row and then collect them all in a row. Good question.
And the whole idea is to keep the sediment intact, and those sediments with their vertical orientation intact.
Good, Kylie.
Is 4 all we've got today?
We have 5.
Oh, 5. We had to do a different configuration of the ROV today, all of the tools that we brought. As Lisa was mentioning earlier, we've got a bunch more samples already on board that we're going to deploy and leave on the seafloor for a while. So we couldn't bring some of our coring tubes because we had to make room for all of our awesome colonization experiments that we will be dropping off at 2 different depths during the dive today. So it's the OET crew here is really awesome. Every day they ask us how we want the vehicle to be set up for our different science objectives, and they help us get them all set up in the right configuration.
We have geologists dominating the science chat.
Yeah, we've got another
thing.
Yeah. We should ask.
Chattering away.
Yeah.
Go ahead, Carly.
They're all very excited about this particular site. Maybe we can ask them what they're hoping to see. But they can't talk back. Oh yeah, I guess that's true. I don't have to type it. Yeah, so Jamie Conrad from USGS in Santa Cruz, where I work, just joined. We've got some other geologists, Mark Legg and Mark Kammerling. What are you guys hoping to see here today? If you can type it in the chat.
Kylie, you want to bump the box in a little, please?
Sure. Sample tray in a bump.
A little more.
A little more.
No way I can reach around it without beating it up. Keep going.
What do you think? How's that?
A little more. That's better. Except for this float.
So I don't think people viewing get a real understanding of how hard this is to do. Just has to reach around the side and look in a couple different camera angles that neither are great. And there's a float in the middle of the view.
It looks like it'd be hard to determine depth perception too. So Mark is saying he hopes to see some very steep slopes and scarps with possible slicken sides to determine if this is a head scarp of landslide or a strike slip fault. Gary, you're going to have to explain some of those words to me.
Or you might be on mute. Not
What?
hearing you.
On mute?
No,
No,
I
we
can
can
hear
hear
you.
Yeah.
I think you might be on mute.
Are you, if you have
no.
SPL off, maybe?
Oh, I know what happened. I have SPL.
Oh, there we go.
Kira, can you explain what a slicken side is?
I'm guessing you just mean smooth rocks along the side, which would be like exposed rock that's been smoothed over time, like we've been seeing, or if it's more like a rock garden or a landslide, you can imagine it would be lots of broken up rock. Maybe if Mark wants to see if I was right. Most these guys are the real structural geologists. Everybody's interested in faulting here, so maybe we'll see some cool evidence of faults along this region. And Mary Wickstein's keeping us in the biology headspace, asking if there's a little hermit crab in the lower left.
I'm not sure what she's looking at. Right
That?
there.
Is that a hermit crab right there? It's hard to tell.
There.
There's
Sounds
something
wicked.
moving here too.
That might be it, I think.
And Jamie's bringing up another geology concept near and dear to Lisa's heart. He recently saw an oil slick and a bubble plume here on the sea surface a few kilometers away, and he's interested if there's other evidence of seafloor seeps. And you've done a lot of biological work near seeps, right, Lisa?
Yeah.
Go ahead,
I
Kylie.
would love to find a seep around here. I wish we'd known the location of that bubble plume.
Yeah.
Maybe Jamie can send us the coordinates, so if we come back to get our colonization experiments, we could
Yeah.
think about about checking it out.
Lisa and her group will be coming back with the Falcor. Hopefully next year, if COVID allows all ships to be planned and gone out.
I hope everything's good by next year.
You never know.
This one's going to be a little out of line since I have
It doesn't matter, really. Does
Roger
it?
that.
I think that it's fine.
Nope.
Anywhere, it's fine.
Rog.
Whatever is easy. And you can just punch it in and pull it out without letting go even. If you can get it in all the way.
Okay, so Mark provided some additional info on slick and slides. I was a little off. When rocks get scratched, that's called a slick and slide, and the direction of those scratches can show you the direction of fault movement. So vertical, large scratches would be a landslide, or horizontal for a strike slip fault action. That makes sense. You've got rocks sliding against each other horizontally, you'll have that kind of space, or if they've been sliding vertically down the slope, you'll have vertical scratch marks.
Geological detective work.
All right. All clear to pull out?
Yep.
Rog. Come a little
Thank
wide
you.
there.
So this was a recore, right? Same one? Yeah.
Looks good.
Roger that. A little wide there, Al.
All right, and this should be NA124-149. Push core 5 with the green tape.
Go ahead, Kylie. Too much slew.
That marker is really taunting.
Yeah, they're always an issue if they're not rubber banded together.
Yeah.
Look at that shrimp.
Oh,
Beautiful.
I love their little legs. They look so silly.
What the people watching aren't seeing is the starboard bio box, which
Yeah.
is where the cores live after they're sampled, taken. And the pilot has to maneuver them into a fairly narrow hole in the quiver.
Yeah.
Does not look easy.
Nice. Cured.
So they position the core over the quiver and let it gravity take it in, and then they usually use the manipulator to punch it down onto a plug that holds the sediment in place.
Reminds me of like putting in golf. Kind of high stakes and quiet. Everybody watches respectfully.
For putty golf?
Well, when you're putting in golf.
Oh,
Like on golf TV when it's all quiet.
I see. Yeah. I meant like the theme parks and things. Go ahead, Kylie, and switch it back.
Switch it off.
Okay, she's going.
We're definitely going to golf clap when you get it in the quiver.
Golf clap. Oh my. Let me change up my angle a bit.
Oh, sounds like they do have the side camera live today. So
What they do?
pay it in. My husband John's been watching diligently every day we're out here. Hey, John. Thanks for letting us know you
Go
can
ahead,
see it.
#1 fan.
Let me reset myself over here real quick.
Got a little bit of information about that possible seep they saw. Apparently it was reported to USGS by the Coast Guard. They thought it could be a natural seep or maybe a shipwreck. And the Nautilus was out here checking around for it recently, but the bubble and the oil slick were gone. And nothing obvious in the MBES data. So who knows? I feel like I can hear it good, no?
I can hear her.
For me, it kind of feels far away.
Okay,
Better.
cool. Thank you.
Go ahead, Kylie. Thank you.
It's got 150.
That was 150.
Correct. NA124-150.
Mary Wickstein tells us that big red shrimp that swam by was a Benthiocides tanneri. Benthiocides. Used to be Benthysimus.
This one's only halfway there. So that's usable for you guys.
Yeah, that should be enough.
You want to record the shrimp?
Yep, I've got it.
Go ahead, Kylie.
Sample.
Thank you. I feel like I'm in there, right? Yeah.
Looks like it. Yep.
Come on. Yeah, I'm in there.
There you go. Asking you shall receive.
That wasn't too fast on the descent there. Excuse me, float. You are. Okay.
Oh, I must have missed the last
Go ahead, Kylie. Thank you.
You're welcome. Gosh, I like that shrimp.
There's that benthioscetes again.
With the cucumber?
No,
Shrimp.
the shrimp.
Oh, the shrimp.
Good, Kylie.
Sample.
Thank you.
So is there like a easy answer for how benthic creatures get like coloration, or why they might have it this deep where there's
You're coming across pretty
no light?
quiet, sorry.
Oops, sorry. Can you hear me?
Yes.
Is there an easy answer for why there is so much like saturation or different colors and different benthic animals down here when there's not really light to be perceived amongst each other to be able to see it?
Well, that's one of the reasons a lot of deep sea creatures are red, is because of the light attenuation coefficient, where red light has I think it's the longest wavelength, so that's the one that's absorbed the most at the top layers of the ocean. So red light doesn't really reach the bottom of the ocean. So then these would be the hardest to see. So they just appear darker.
Yep. That's really cool.
I mean, there is light, but it's bioluminescence
Right.
down here, and I have no clue whether bioluminescence can reveal color.
Yeah, me neither.
This one's not wanting to go. Go, buddy, go.
It all looks sort of greenish, fluorescent to us, but blue, green.
Yeah, that's true. We tend to see lots of purples and reds down
I'm not very happy with those.
Might make a note that some sediment might have. It's unclear if that came out from the top or the bottom.
Peter Franks would be so happy. That was actually one of the questions on our
Midterm?
midterm. Yeah.
Really?
Yeah.
About why organisms look red and black
A
in
lot
the
of
deep sea?
Yeah.
Kylie, you want to bump out the starboard bio more now, please?
Sure.
And tell him you got to use it.
Yeah.
That's great there. Thank you.
No problem. Back to dive?
Yeah, please.
No problem.
So that was sample NA124-152 for push core 2.
Go ahead, Kylie. Thank you.
Mary Wickstein suggesting that the red pigments, like the ones we saw on the shrimp, could have a role in intermediary metabolism,
What?
could aid
is
in
that?
respiration. But their study she's citing was over 40 years old.
It
I
was,
mean, we have learned that deep sea shrimp have all sorts of visual capabilities just on their exoskeleton and their, not even just their eyes. I
Great.
recall there was a paper that came out recently about that.
You said intermediary, what was the next word?
Metabolism.
Oh, metabolism.
That
Intermediary.
to aid in respiration. I mean, normally red organisms have hemoglobin, but shrimp also, crustaceans have other red pigments.
Oh, it's just like everything went, sorry.
Al, it's hard to hear you. Your mic is close, but I don't think I have your volume down. I have you have +5 dB.
Okay, Kylie, go ahead and push in that box there.
Sample tray in.
Are we worried about the float
Stand by.
doing anything?
Yeah. Shove this guy back down in there.
I don't think it'll stay.
Yeah, that's true. All right, Kylie, I'm going to kill the verts there.
I mean, it is possible we could take the float out and
Go ahead
hold
and close
it
Go ahead and close it, Kylie.
if it's a
Close it.
problem.
I think it'll be all right.
Should be safe there, right?
Yeah, I think we had it like that on the last time we deployed the colonization experiments.
Yeah, we'll just keep an eye on it. Cool.
All right. Nice job, Jess.
Way to
Yeah,
go.
sorry for the delay.
Thank you. That was great.
Great job.
All right,
Let's go find some rocks.
Going over. Thank you. Look at all that schmoo going on here. Wow.
Apocalyptic.
That was just from disabling and re-enabling thrusters. Like soup. Let's get out of this. Gross.
Callie, this is a level check. Oh, wow. I can see on my meters I'm very low.
All right. Aaron, I am ready for you.
All right. So next waypoint AH is at 090. It's 185 meters. Just want to note that we are at 1100. So I don't know what your tolerance is for how far away from that you want to get. I know you want to land on rock, but how much change in depth are you willing to sacrifice?
Well, probably no more than 100 meters, but I'm guessing we'll be okay.
Okay, we'll keep an eye on that then. All right, so we're going to be moving 090.
090, Reg. I'm just doing a car wash if this looks crazy. I promise it's intentional. There's a bunch of sediment that'll otherwise obscure viz.
Reg nav.
Okay. Okay, got it.
We get a 20 meter step bearing 090.
Sorry, what's the heading again?
090.
090. Okay.
You're already there.
Yeah, you're pulling on me. Do
I see a rock.
you want less delta since we're stretched out?
No worries. We're good.
All right, so eyes out for rock to deploy these guys on.
Reg.
And for a rock to pick up.
I see a fish, maybe.
Is that a little thorny head down there?
Or there, it's a rock. I can't tell. There's a thorny head next to the big flat slab down there.
Oh, yeah. Let's see.
But I'm looking at what's straight ahead. Is that a fish?
Looks like some form of flatfish there.
A big old sole.
Al, if you want to do a partial on that fish there, so you don't see all the Campbell's soup next to us.
Sure. We're waiting for the
Partial
ship.
on the fish.
Are we waiting for it to clear, or are we just...
No, they're just waiting for the ship move to... So she's pretty far ahead in Argus's view. So as the ship gets moving, she doesn't want to pull out of that view. So they're just... entertaining us while we wait for the ship motion to come through.
Do a dance.
It's a nice rock.
Hard to tell, but it looks kind of grayish.
All right, we'll go ahead and come wide there, Al.
Is that a little bright zone on the bottom right of that rock too, it look like?
The white thing or the encrusting?
The white thing, yeah.
You can take another look. Go ahead, Al.
Bridge nav, another step, same bearing.
That's a coral.
Coral, yeah.
It's
Correct,
like
thanks.
a solitary coral. Big coral head, actually.
A brittle star on the left. I like how
Look
fast
at
it's
it
moving.
go. Running.
Running across there, yeah.
They're so funny.
It's like the breaststroke.
It is. Sediment breaststroke.
That looks like a big soul of some sort.
Just, you want a junk zoom on this?
Sure thing.
Get a
There we are.
close-up of this nice fish.
Go ahead
What
there,
size?
Al.
Coloring on it.
25 centimeters, maybe.
Hello, friend.
Stand by.
I don't
One
think
thing
we
that
have
would
any
help.
fish people on who can tell us which fish this is.
Gonna get myself all sorted out with my joy gains. Flying like a mad person.
Looks like Embasictis with the spots.
Oh yeah, it
Yeah.
does. Genus Embasictes.
What is that to the left? It looks like a piece of garbage or something because of the pattern on it.
Oh, a grenade.
Yeah.
what I
I mean,
was
that's what I
thinking.
thought, but I wasn't going to say it.
Let's not get too close.
Yeah, let me write that down. Yeah, looks like genus Embasictes.
Deep sea soul, Erica Burton tells us. Embasictes bathybius.
All right, come on, please, Al. Start moving.
So if you're looking up there, when you see that she's starting to come out of view, then you know the ship has got some motion on because Argus is being pulled over.
Thank you.
It can be cheated too with the angle of my camera if I'm looking straight down or out ahead. If I'm looking out ahead, she's way stretched. We are way stretched. If she pulls on me, I'll just change my heading and I'll lose view of Herc.
Bridge nav, another step, same bearing.
Nice big sediment plain here.
What is that?
Pepsi. Go ahead, Al, and push on in on the trash.
That's an
Oh,
old
old
what
school.
kind of
can.
species is that?
That's circa 1975.
Hepsibius.
Cypsius.
Pull wide, please.
Hepsibius.
Too bad.
Hope it's a species that goes extinct in that form.
Ooh, different.
It's a
Okay, there's
dark color.
our dark purple urchin. I've forgotten the name of that one.
Is that not
Somebody-
Panickia still?
No,
I've
I
seen
don't
them
think
together.
so.
I haven't seen two different kinds of cucumbers.
I'll have to look at the Panickias are usually paler, but I don't know if they have another
There's a
form.
paler one right to its left, yeah.
Right.
They look pretty similar except the coloration.
Maybe they're two color morphs.
I wonder if that has something to do with diet or
Probably genetics.
genetics. Wow.
A rock.
Interesting. Big rock.
I'll come over in front of you there, Kylie. No worries.
Thank you. Yeah.
Or a bit deeper, which is where we had a little bit harder time.
Relatively high oxygen
Bridge nav,
saturation
another
too.
step, same bearing.
True.
Or 4.5.
Well, this is our high oxygen deployment down here. In a
Some more
relative way.
thorny heads.
It's like tarantulas.
That
like
is
a
a crab. Maybe a tanner crab?
The underwater crab or underwater
Lithoted.
spiders.
Spider crab. That's why they're called spider crab.
I was looking at a paper that had some old deep sea images in black and white. So it looks like an old movie with the spider crabs looking kind of, I don't know, like an old horror movie.
So the sea cucumbers here look white-ish, some of them,
Or dark purple.
or covered in sediment.
What's this?
It's that shiny thing up top.
It's like, gonna zoom in on this guy
Maybe
here.
a sponge skeleton on the surface.
Looks like
Bit
a
of
or a
garbage.
Is that a trash
or a
bag?
bag.
bag,
Yeah,
yeah.
an old
There's a lot of
bag.
trash at the site.
Well, we're only 40 miles from shore,
Some right
and
there.
that's one of the reasons.
Yeah, looks like another can here.
I'm sure there's recreational fisheries and all sorts of people presence
That's
in the
unfortunate.
vicinity and islands.
Another one of those. Is that sponge with the anemone on it? The symbiotic relationship?
Yeah, but the sponge is not there. Just the dead stalk and the anemone.
That's the fiber optic sponge.
Go ahead, Al.
There's a whole series of hydroids and other things living on it as well.
A little wide, please. Thank you. Full wide.
Jess, would you like full wide to be past the junk?
Yeah, past the junk.
Ridge nav, another step, same bearing.
For those people just joining us, we're headed across the sediment into what we hope will be a rocky slope, looking for a place to deploy a series of hard substrate colonization experiments.
And maybe pick up our first rock of the dive once that operation is successfully deployed.
We're actually pretty floaty right now, flying-wise, so if you guys want to pick up more rocks, larger rocks, we're always happy to do that.
Okay,
Thank you.
Make us fly better.
we can add a porch rock or two if we want.
That's true.
Yeah. Yeah, porch rocks.
That's a new thing for us, porch rocks.
And as we get into these rocky areas, the geologists are giving us all kinds of interesting fault features to look for. Yeah, sorry if you guys notice us sounding far away or hard to hear in the mics today. We're all wearing our masks for our COVID protocol on board, keeping each other safe. So it's fun to manage mics and masks, and we're all amateurs here in the back row.